So I'm a senior consultant in obstetrics at Karolinska University Hospital, and I'm a docent at Karolinska Institutet. And so my talk is not about intrapartum, not even hypoxia or acidemia detection. What is this? I'll just very briefly explain why this happened. When I just finished my residency years and was walking one day from our hospital buildings toward our administrative building where we had our offices with doctors, I had a sliding door experience, or more of a non-functional door experience, which changed my whole career, you could say. So this door was not working. You had to wave a lot in front of the automatic door opener to make it open. So I stood there waving, and a senior colleague had his office on the other side in this corridor, and he thought I was very happy to see him waving like this. And he was in search of a doctoral student and invited me to discuss his project. And that resulted in my thesis, lactate as an early marker of intrapartum fetal hypoxia. And my continued research has 3 traces, of which 2 I collaborate with my colleague at the Royal School of Technology, Dr. Farad, who is also here with me. So we have the intrapartum fetal heart rate monitoring, traditional visual assessment, and computerized CTG. And I've also continued with the fetal lactate as an adjunct tool to CTG, and I collaborate with another research group at the KTH also. And the third track is antenatal fetal monitoring and alternative areas besides detecting or searching for the chronic hypoxic growth-restricted fetuses. Is there other uses for antenatal, where antenatal CTG could be used? And 1 to 3% of pregnancies are complicated by preterm, pre-labor rupture of membranes, pPROM. And pPROM causes approximately 1/3 of preterm births. And one main complication of pPROM is intra-amniotic infection. And the intra-amniotic infection increases the risk of very severe both maternal and neonatal complications, such as early onset neonatal sepsis, EONS, which I will refer to. So our monitoring tools, this is quite a large patient group at our antenatal ward, the pPROM women who are admitted for weeks and weeks if the water breaks at extreme premature period of time. And we monitor them by clinical assessment, temperature, amniotic fluid, abdominal palpation to see, is there an evolving infection? Because otherwise we want to wait and let the fetus continue to mature its organs intrauterine. We take blood tests, C-reactive protein and white blood count, and we monitor the fetus, of course, for tachycardia or etc. All these tools have been shown to have a poor diagnostic and prognostic performance regarding intra-amniotic infection and early onset neonatal sepsis. This is only one example of the publications that have studied the performance of all these clinical and laboratory tools. So as clinicians, we stand there weighing the balances of risks in week 24, in week 27 or 29. If we wait, will that cause life-threatening infection in the fetus newborn? Or if I cause an iatrogenic extreme preterm or moderate preterm birth, will I increase the risk for this newborn to have intracerebral hemorrhage or other complications due to immature organ systems? So it's a delicate balance for us clinicians. So what about the short-term variation of the fetal heart rate? It's affected by metabolic acidemia, that's already known, and that's how it's used today as part of the Dose-Redman algorithm and other parts. And it's increasing by gestational age. It is decreased during fetal deep sleep, and decreased when the fetus is tachycardic, maternal diabetes and maternal smoking are affecting the short-term variation, and some medications. Most of all, we think about the corticosteroids. What about the effect of an infection, of an intra-amniotic infection on the STV? We only found it's very scarcely studied, although we discuss a lot about inflammation effects on the fetal heart rate, as was done on Monday. But publications on this particular thing, I came across in 2016 on a maternal fetal medicine meeting in the US, an abstract on a very small cohort or more case control study of 10 cases with chorioamnionitis, histologically validated, and I think 13 controls. But it was presented as a cohort study. However, they showed a lower short-term variation among those women who had a chorioamnionitis. This could imply a possible effect of the infection on short-term variation and could possibly give us an additive tool to monitor and survey these pregnancies, these high-risk pregnancies. So we wanted to investigate the association between the exposure to intra-amniotic infection and the outcome, short-term variation of the fetal heart rate. And we extracted all the pPROMs between week 24 and 34 in the 5 hospitals in the largest region in Sweden, the Stockholm region. And the exposure was the intra-amniotic infection, and the outcome was the last calculated mean STV prior to labor, start of labor, or prior to cesarean birth. How to define this infection? We don't perform amniocentesis because then we can introduce infection. There are the risks with taking an amniocentesis to culture the amniotic fluid, they are too high. Well, to use the chorioamnionitis, as the colleague van der Broek had done, we didn't think it was so good because chorioamnionitis, even if the histological validated ones, they are very common, and they can be seen without a positive microbiology and biochemical markers for inflammation. And the prevalence of chorioamnionitis at the time of delivery is very high among women with pPROM. Almost half of them have it. So using an outcome that happens in half of the population, it's not of such big use. So we used quite a controversial proxy for the exposure, the early onset neonatal sepsis. Because it is caused by vertically transmitted pathogens and is strongly associated with intra-amniotic infection. And also most preterms with early onset neonatal sepsis, they are born to mothers with intra-amniotic infection. But you have to think about it. We have a proxy, nothing happens, no, another one. We have a proxy for the exposure, which is actually set. The proxy is here after the baby is born, after the outcome. But bear with me, you'll see the benefits of it. So we started with 922 women during 7 years in the Stockholm region with pPROM between these gestational age weeks. Why we stopped at 33 plus 6 weeks was just from a clinical point of view, because for me as an obstetrician to decide upon delivery, induction or cesarean after 34 weeks, when the baby is so mature that it doesn't need the corticosteroids anymore, it's not a difficult decision. The difficult for me as a clinician is in these extreme premature and moderate premature weeks, so that's why we chose this. But it really had some back side effects. this design. However, we excluded some due to we didn't want to include labor. So the pPROMs within 12 hours from birth could be part of the start of labor. And we didn't want to interfere with labor effects on the short-term variation as possibly hypoxia, acidemia. And insufficient data, intrauterine fetal death, and a couple of others. So we had 648 patients, and 25 of these had intra-amniotic infection with this sepsis, neonatal sepsis diagnosis, whereof 11 had a positive culture, and 622 had no intra-amniotic infection. Before analyzing the data, we drew up a directed acyclic graph to decide what is to be assessed as a confounder and what should we control for. And we ended up with only diabetes and smoking as confounders, and a lot of other, which quite a lot of reviewers wanted us to control for, such as baseline and gestational length. We assessed them, and we discussed this a lot among within the research group. We clearly assessed them as either mediators, we didn't want to over-adjust, or not affecting both the outcome and the exposure, but only one of them. The mean STV was calculated in the last 30 minutes with my colleague Farad's help. And we used the traditional Dose-Redman algorithm. And this was, as I said, we excluded labor traces and used the last 30 minutes prior to either start of labor or birth with cesarean without contractions. We excluded those with a loss of signal loss more than 10 minutes, and Dr. Farad used MATLAB for the analysis. Different hospitals used a few different machines, so it was Sonicade, Avalon, or EDAN. The women differed between those with and without infection only with in smoking and diabetes. Otherwise, there were no difference. And the neonates, they differed in APGAR scores at 1 and 5 minutes. Otherwise, no. And the main results are here. The mean STV in fetuses, it was most pedagogic to show the relative difference in short-term variation. So the highest, the minus 23.5% decrease is unadjusted, and it was most pronounced for the confirmed, with the blood culture confirmed eons. And when we adjusted for smoking and diabetes, as you see in the middle, the minus 23.4, did not much happened. And although I just explained why we didn't want to over-adjust, we didn't want to see duration between pPROM and birth and baseline and gender as confounders, but mediators, we still saw the questions from reviewers, etc., coming. So we did that calculation anyway, and as you see, the difference disappears. I'll come back to that. And to be able to although we considered chorioamnionitis as a quite non-functional proxy for intra-amniotic infection, we still wanted to be able to compare our results with the only other study that we knew of. So we had the information among some of the in the population of histological chorioamnionitis and also fetal inflammatory response as is diagnosed in the umbilical cord. And we could see that we had a very similar result as Dr. van der Broek, with a significant decrease of minus 16%, both unadjusted and adjusted. So we concluded that fetuses in pPROM pregnancies complicated by intra-amniotic infection has a lower short-term variation compared to pPROM fetuses without an infection intrauterine. And with better diagnostic tools for intra-amniotic infection, we can decrease both underdiagnosis with the risk of having severe infections in the neonate, risk of death or lifelong sequelae, and also risk of overdiagnose with us causing iatrogenic extreme preterm or moderate preterm birth. So this might lead to improved both maternal and neonatal outcome. So this was the tricky part with using a somewhat different kind of proxy for the exposure and the outcome. We have now gone further and looked into the longitudinal change in short-term variation in these pregnancies with and without infection. And we have assessed the longitudinal development of the mean short-term variation at the baseline frequency, and we visualize it using locally estimated scatter plot smoothing curves with the help of a statistician, which is weighed against the inverse of the total number of measurements points for each fetus. Since this is a retrospective observational study, we cannot decide how many times the women have gone through CTG monitoring. So we have the traces we have. And we can there show that the baseline does not explain the entire effect that we saw on the short-term variation very clearly and beautiful. But I cannot share those pictures, unfortunately. We wanted to perform a prediction model, but due to our study design, we came to the conclusion we cannot do that, but we are performing a risk factor study. There are several risk factor studies that is comparing lab estimates, CRP, white blood count cells, clinical pulse temperature, etc. But we want to compare the short-term variation with the traditional risk factors to see if we can add a diagnostic tool. This is the research team and funding, and the PhD student Tina Birgisdotter at the bottom has done very thorough work with this, but she couldn't be here either due to small children and in the middle of summer. And we have also a neonatology professor in the team besides Dr. Farad and my postdocs. And in my other research track, I have other colleagues from KTH and other obstetric colleagues. One I want to mention Professor Beskren, who made the groundbreaking lactate studies in the 80s and 90s and set the cutoff for the clinical use of fetal blood scalp sampling with lactate analysis. So I think I'm running late. Thank you for listening.